Hello everyone, this is Prashant from Government College of Pharmacy, Bengaluru. In this video, I will be discussing about the determination of protein structures, mainly by computational methods. And before going to the determination of protein structure, I will briefly discuss about the proteins and how the protein structures are formed and different types of protein structures. We all know that proteins are the building blocks of human body, and these proteins are formed by the polymers of the polypeptides, which are basically known as amino acids. And these amino acids, during condensation reactions, it loses a water molecule by which it forms a bond with another amino acid, and this process goes on and to form a peptide chain. And this peptide chain will undergoes a conformation change which forms a protein. And this protein, it consists of either hydrogen bonding, van der Waals force, and ionic interaction and hydrophobic packing. And these non-covalent interactions which will help the protein to form say special conformations, and these conformations will be unique for that particular protein. And this special conformations helps in the helps the protein to perform their biological functions. And this alternative structure is also referred to as a different conformation isomers, simply conformations. And the transition between these conformations is known as conformation changes. Basically, there are four types of protein structure, and they are primary, secondary, tertiary, and quaternary. In primary, it consists of a simple linear arrangement of amino acid sequences, and which is formed by the sequence of amino acids and is a straight chain. Whereas in secondary protein structure, it forms a binding pattern of the amino, hydrogen, and carboxyl oxygen groups between the amino acid sequences through the peptide backbone. And the secondary protein structure are basically of two types, which is alpha helix and beta strand. This alpha helix, it consists of a coiled structure, and it consists of 3 to 6 amino acids per one turn. Whereas this beta sheet are straight chain, it consists of upward or downward ring which is arranged parallel to each other. And this alpha and beta strand, it forms a globular strand which is of 3D structure to form a tertiary structures of the protein. And this fold is initiated by hydrophobic bond, disulfide bond, and salt bridge, and also hydrogen bond. And quaternary structure, which is built up of dimeric or multimeric molecules stabilized by the non-covalent bond. The structural annotation of a protein is key understanding towards the function of a protein. So now moving on to the prediction of protein structure, there are basically two types. One is experimental based, another one is computational based. In experimental based, we use NMR, X-ray, or electron microscopy, whereas in computational based, we use homology, threading, or ab initio methods for the protein structure determination. And basically both types of protein structure determination helps in the determination of three-dimensional models of the protein, and which is based on the similarity of the amino acids of these proteins, which are similar to another protein with those dimension structure which is already known. And whether it is computational or experimental based, this both protein structure determination begins with the decomposition of the structure. It means it will break down of the structure into one or more basic units, which are called as structural domains. And these domains undergo domain shuffling when they reappear in different combinations, hence they will perform different functions. And the most common definitions of this domain is referred to as structural domains, and these structural domains are the units of that protein structure that which are compact, and these are stable, and it consists of hydrophobic core, and it can fold independently without depending on the rest of the protein, and it occurs in combination with different domains and perform a specific function. And I told that there are various techniques to determine the protein structure, but the experimental structures such as X-ray, NMR, they have some limitations, and these limitations are that they are very time consuming and they are expensive. And other types of limitations are that some proteins are very difficult to crystallize, and hence they are very difficult to identify by X-ray crystallography. And NMR spectroscopy is only applicable for proteins with less than 300 amino acid residues. So hence, because of all these limitations, the most widely used and very time consuming, and we can perform it at very low cost as computational methods of protein structure determination. And there are basically three types: one is homology modeling and threading modeling, and another one is ab initio method. This homology and threading model is mainly based on knowledge based. That is, it determines the protein structure with comparing it with already available protein structure based on its similarity. Whereas ab initio methods is known as physical based approach, and it is non-template based basically. That is, it does not identify the protein structure based on identifying the proteins which is already known. That is, it mainly determines the protein structure with the help of the physical base such as steric hindrance and torsional energy and pi value, psi value, etc. This homology modeling it was published in 1969 by David Phillips Brown and their co-workers, and it is also known as comparative modeling. And it is defined as the identification of one or more protein which is similar to the structure of the target protein sequence. And this homology modeling is mainly based on the principle that we all know the phrase which is known as similar structures have similar functions. In the same way, this homology modeling is based on the same principle that similar amino acid sequence may have the same function, same structures. And this structure of these proteins are similar in the sense that amino acid residues with identical physicochemical properties occupy the same position in homologous proteins. And this homology modeling it mainly consists of the following steps. And the first step is template recognition and alignment correction, and backbone generation, loop modeling, side chain modeling, model optimization, and model validation. And I will discuss each step in further slides. Now, the first stage is template recognition and initial alignment. Now we have some unknown protein sequence, and we have to compare it with another protein sequence. Hence, we have to recognize the target, recognize the template protein, and this is done by with the help of the searching of the PDB. PDB means the protein data bank, and which consists of a number of proteins and the number of protein structures. And first, in template alignment, we will first search the PDB for homologous protein, which consists of determined structures. And this PDB will use the tools such as BLAST and FASTA search. And BLAST search it is a search algorithm which can compare our query sequence with the sequence present in a library or database. And it gives us the sequence which match our query sequence. It can search amino acid sequence of protein nucleotides of DNA and RNA sequences. Whereas in FASTA, it is a software which is primarily used for DNA and protein sequence alignment. If this FASTA is used for DNA, it is known as FASTD, and if it is used for the protein sequence alignment, it is called as FASTP, and it has a high speed of execution. First, it marks the potential matches using the pattern of word hits and word to word matches of a given end. It is also famous for its FASTA format, which has become widely used in the bioinformatics domain. And the next step is alignment correction. Now we have aligned the target sequence and the template sequence, and there may be a mismatching between this, and hence this alignment correction is mainly useful. And the alignment of two sequences of the two proteins where the percentage sequence identity is low can be difficult, and one can then use the other sequence present from the homologous proteins to find the solution. For example, it is nearly impossible for the sequence LT to be aligned with the sequence YA. Hence, we find the third sequence TY, which can be easily aligned with both of them. And hence, this alignment correction is very important. And another thing is in template recognition, we will study how much percentage the similarities between the template and the target protein. If the similarity is less than 20, and it comes under the midnight zone. If the similarity is between 20 to 40%, the target protein comes under the twilight zone, and if it is more than 40%, and it comes under the safe zone. And most of the proteins, it should be above 60% similarity to be useful for the homology model. And the next step is loop modeling. And after the sequence alignment step, due to insertion and deletion, there are one or more loopholes which are generated in the protein structure, and this leads to the gap. And this loop modeling technique is used to fill this gap, but is less accurate. And there are mainly two types of techniques which are used to fill these gaps. That is database searching method and ab initio method. In database searching method, this involves going through the database of known protein structure and finding loops, and then superimposing these loops onto the two-step region of the target proteins of the target protein. And this ab initio method, it various loops are generated randomly, and then it searches a loop in those loops which are reasonably low energy. And the next step is side chain modeling. It is important because it is used to evaluate the protein ligand interactions at the active sites and also the protein protein interactions at the contact interface. By searching every possible conformation for every torsion angle of the side chain, we can select the one which has the lowest interaction energy with neighboring atoms. And once after this side chain modeling, next we will do model optimization. Model optimization is done so that the overall conformation of the molecule has lowest possible energy potential. This is done by adjusting the relative position of the atoms, and this leads to a better chance of finding the true structure. And the energy is calculated by combining all the following energies such as stretching energy, bending energy, torsion energy, and non-bonded interaction energy. And the next step is model validation. And every homology model contains errors, and the main error occurs by either two types. And the first error occurs if the sequence identity is more than 90% or less than 30% of the template and the target protein. If the sequence identity is more than 90%, then it can be compared with the structures which are determined through crystallography. Else, if it is less than 30%, then major error can occur. And the second major reason for errors is the errors presence in the template itself. The advantages of this homology model is it can find the location of alpha carbons residues inside the folded protein, and it can hypothesize structure functions relationship, and mutagenesis experiment can be guided using this type of homology modeling. And they use active sites, binding pockets, and the ligands can be identified using the positions of the conserved regions of the protein surface. And the disadvantages of this technique is the side chain position and predicting conformations of insertion or deletion are not possible with homology modeling. And because it uses other types such as I told database searching and ab initio models are used to fill the loopholes, and hence it can not be done with using homology modeling alone. And these homology models are only useful for drug designing and development process if the sequence identity of a target protein is more than 70% with the template. Because template with less than 70% identity cannot be used in modeling and ligand docking studies, which is necessary for this scenario. And the error sources in homology modeling is either it occurs through incorrect sequence alignment, incorrect choice of template may happen, especially for multidomain proteins, and the loop regions can be built incorrectly. Normally, the servers build the loops automatically. And the person doing the modeling can also make some errors, but this type of errors can be hard to predict, and it can include any kind of errors. And one way to minimize this kind of errors is to have basic knowledge about the principles of protein structures. And there can also be errors present in the template itself. This makes this type of errors difficult to deal with. And these are the commonly used homology modeling softwares are INT Ford, Raptor X, Biscuit, Modeler, and Sys model. So the next type of computational methods are threading model, or also called as fold recognition model, and it is a process of comparing the target protein sequence with a library of templates, and the templates consist of the protein structure and fold data of various proteins. This type of modeling is done when the fold of a protein match the folds of a protein whose structure is also known, but the structures of the proteins are not homologous. That is, it is not based on the amino acid sequence, it is based on the folding pattern of the protein with another protein. And we all know that folding of the protein is a physical process in which a random coil folds into its characteristic and 3D structure from a polypeptide. It matches a protein sequence to a fold library using so-called threading algorithm that assigns each amino acid to a position on three-dimensional structure for that particular fold class, which is available in protein drug bank. And this protein structure, it cleaves the complete, this threading model, it cleaves the complete protein into libraries of the protein, and based on that particular folding pattern, it identifies that this folding, since the folding is similar with the template and target, this target sequence may contain the same amino acid sequence of that of template strand. And the steps involved is first is construction of structure template database. In this, we will select the protein structure from protein PDB. When selecting the protein structure, we need to make sure that we avoid protein structures which have high sequence similarity, because we are mainly concerned with high folding similarity, not sequence similarity. And next step is the design of the scoring function. And mutation potential, pairwise potential, secondary structure compatibilities, and gap penalties are the main things a scoring function should consist of. A scoring function should measure the fitness between the templates and the target sequence. Next is threading alignment. The scoring function has to be optimized so that the target sequence can be aligned with each of the structured templates in threading with structure prediction program, which take into the account the pairwise contact potential. And threading last step is threading prediction. Choose a threading alignment that is statistically likely as the threading protein. Structure model is constructed by using a selected structural template and placing the backbone atoms of the target sequence on the structural templates aligned backbone proteins. The advantages of this threading model is it is used successful and it is good for proteins with less than 100 residues. And the disadvantages is that it assumes that the current structural library consists of all possible conformations that could possibly be deciphered experimentally. Less than 30% of the predicted first are true remote homologs. High computational cost is involved to screen a library of thousands of possible folds. And energy functions are simplified for efficient calculations and results are therefore compromised. And the softwares which are commonly used for threading models are HHSpread, RaptorX, FireN, Fire2, Falcon, and Mustard. And the last one is ab initio method for protein structure determination. It means from the beginning, and this method predicts the structure of protein from the scratch using sequence information by applying Newtonian force to achieve a thermodynamic stable native-like state. It is very useful in situations where the query sequence does not have any suitable applicants in the protein structure library for either homology or threading model. While homology modeling and threading modeling requires knowledge of known structure, ab initio prediction has no such limitation because it mainly uses the physical. It is mainly based on the physical structure of that protein. It starts with the assumption that the necessary structure of the protein is at the global free energy minimum. This method has assimilated information obtained from known structures and is based on picture of a protein folding in which short segments of the protein chain flicker between the different local structure are consistent with their local sequence. And folding to the native state occurs with those local segments are rotated such that low free energy interactions are made with throughout the protein. And the software which is used for ab initio methods are Rosetta, etc. And the overall applications and limitations of this computational methods are: each method discussed provide structural details to different extent. Homology modeling can provide atomic levels of the target protein, whereas threading can judge only the fold of the protein. And high and medium level homology model with sequence identity less than 30% are useful in refining functional protein predictions such as ligand binding. Low accuracy models of many of the ribosomal proteins were helpful in building the molecular model for whole yeast ribosome. Folds predicted by threading could be used in supporting site-directed mutagenesis experiment, designing of stable crystallizable variants, and in refining NMR structures. The accuracy and acceptability of models produced by ab initio methods are in general of lower accuracy compared to models obtained from either homology or threading. In predicting functional relationships from structural similarity and for identification of patches of conserved surface residues, this computational methods of protein structure determination is very useful. So this is the overall computational methods for the protein structure determination. First, we will have the unknown sequence, and this unknown sequence of the proteins, we will find this proteins in the database, either PDB or another software, and then we will make alignment between the target and the template and target protein and the template protein. And if the alignment is more than 40 or as I said 60%, then we will go with homology model and we will find a 3D protein model. And if there is target percentage alignment is less than 40%, then we will make secondary structure protein predictions that such as fold recognition or breaking down the protein into libraries of the protein, so that we will determine the protein fold for that particular sequence. And if the sequence structure alignment is plausible, we can again we will build up that uh, all the libraries of the proteins, and we'll go with homology modeling, and then we'll find the 3D modeling. If the predicted food is also not available, then we will use ab initio structure prediction, where it forms the physical. It is used to identify the protein structure based on its physical properties such as torsion energy, rotamer energy, and pi and psi value, etc. And this is all about today's topic. And thank you.